My name is Utyo Basu. I'm an immunologist. I work at Columbia University. My lab focuses on immunology and how that affects in curing cancer. Part of my laboratory is to understand how the immune system works, especially a type of cells called B cells or B lymphocytes. And these cells are known to make antibodies. We understand what antibodies do. They identify different antigens or foreign pathogens that invade our system and remove them. But what we have learned from many other people is that B cells unfortunately is also a place where there's a lot of initiation of cancer. There are many types of cancers that we in general call lymphomas and leukemias that occur because B cells outgrow the requirement in our body and then they become masses which are known as lymphomas. What I've been interested in is how B cells functions normally, how they make antibodies, how non-coding RNA controls the genome and expression of many proteins, and how B cells make antibodies and in that process sometimes lead to cancer. We don't understand what 95% of our genome does because that's the non-coding region of our genome that is a total black box in science. And in this application, what we are trying to investigate is how the non-coding transcriptome has a role in cancer initiation, and the prize is allowing us to answer these questions. There is a lot of emphasis on curing cancer and after a patient has a cancer and how to intervene and treat and cure. But I think a major step will be if you can predict some people who are susceptible to cancer. And what we understand until now from research, we think the non-coding RNA expression in two different cells, a normal cell and a cancer cell, is slightly different. And we think we have the technology to find that. These subtle but clear changes can be amazingly consistent between patients or animal models when we're doing experiments. Recently, from many laboratories have indicated that malaria infection can lead to the very aggressive form of lymphoma, but how that happens is a big black box. We don't understand how invasion of our immune system by a parasite can lead to affecting B cells and leading to cancer. We are really interested to figure out how we can intervene in these events so that we can prevent cancer. We are trying to recreate cancer through infection in mice. And then we want to ask the question why this happened at a molecular level and how that can be stopped.